Hello, and welcome to Study Hub Pro. Today, we're diving into the really cool world of genetics. We're going to learn how to predict the traits of offspring, almost like we're looking into a biological crystal ball. And seriously, by the end of this, you'll know exactly how to use it too. So, this is the big question, right? How can we possibly look at two parents and make a really good guess about what their kids will be like, genetically speaking? It might seem like it's all just a game of chance, but it's not. Genetics gives us this amazing toolkit with really clear rules to figure it all out. So here's our game plan for today. We're going to start with the essential vocabulary, the lingo. Then we'll build up our skills step by step from crossing just a single trait all the way up to 3. And then we'll wrap it all up with a super handy toolkit that you can use for pretty much any genetic problem. Okay, first things first, we have to learn to speak the language of genetics. Getting these key terms down is the absolute foundation for everything we're going to do next, so let's dive in. Let's kick things off with the word allele. So think of a gene as a category, like hair type. An allele is just a specific option within that category. So for our cats here, the gene for hair type has 2 versions we're looking at. One for short hair, which we'll represent with a big S, and one for long hair, a little s. Simple as that. Now this one is absolutely critical. Genotype versus phenotype. The genotype is the internal genetic code. The actual combination of alleles an organism has, like SS, SS, or SS. The phenotype, on the other hand, is what you can actually see, the physical result of that code, like a cat having short hair. So genotype is the blueprint, phenotype is the building. Okay, next up, homozygous and heterozygous. It sounds complicated, but it's not. If an organism has 2 identical alleles for a trait, like 2 for short hair, SS, or 2 for long hair, SS, it's homozygous. If it's got 2 different alleles, like 1 of each, SS, then it's heterozygous. See? Not so bad. So, what happens when you get a heterozygous pair like SS? Well, that's where dominant and recessive alleles come into play. The dominant one, the capital S, is kind of the loud one. It's the one that always shows up in the phenotype. The recessive allele, the lowercase s, is quieter. It gets masked unless you have 2 copies of it, like in the SS genotype. All right, we've got the lingo down. So now we can actually do our first genetic cross. This is called a monohybrid cross, and it's where we track the inheritance of just one single trait. This is our foundational skill. And you know what? There's a simple 4-step process for this every single time. First, figure out the parents' genotypes. Second, figure out the possible gametes, the sperm and egg cells, that each parent could make. Third, we map out what their direct kids, the F1 generation, will look like genetically. And finally, we can cross those kids to predict the traits of the grandkids, the F2 generation. Let's put this into action with a fantastic tool called a Punnett square. We're crossing 2 heterozygous cats, both big S, little s. We put the possible gametes from one parent across the top, an S, and the others down the side. Then you just fill in the boxes, and it shows you all the possible genotypes for their kittens. 1 SS, 2 big S, little s, and 1 SS. And this chart illustrates the result perfectly. Remember, both the SS and the SS genotypes give a short hair phenotype because that dominant S allele takes over. So we've got 3 boxes that result in short-haired kittens. Only that one little SS box gives us a long-haired cat. And that's how we get the classic 3-to-1 phenotypic ratio. Okay, let's try a real-world problem. Mr. and Mrs. Smith both have widow's peaks, which is a dominant trait. But here's the twist. They have a kid without one. So think about it. The only way their child could not have a widow's peak is if that child has 2 recessive alleles. And for that to happen, both parents had to have a recessive allele to pass on. That tells us they're both heterozygous. Their cross is exactly like our cat example, which means there's a 3 in 4 or 75% chance any future kids will also have a widow's peak. Okay, you've totally mastered tracking 1 trait. So let's level up and try tracking 2 at the same time. This is called a dihybrid cross. Yeah, things get a little more complex, but trust me, the logic underneath is exactly the same. So the key idea here is something called the law of independent assortment. It basically just means that the alleles for different traits get sorted out on their own. The gene for a pea plant's height doesn't really care about the gene for its seed color. They're inherited separately. And because of this, we get way more possible combinations, which is why our little Punnett square suddenly blows up to 16 boxes. And here's the beautiful results of all that complexity. When you cross two parents that are heterozygous for both traits, this predictable 9:3 to 3:1 ratio always pops out. You'll get 9 offspring with both dominant traits, 3 with the first dominant and second recessive, 3 with the opposite, and just 1 lonely offspring showing both recessive traits. It's like clockwork.
So, you know what's coming next, right? What happens when we push it even further and track 3 traits at once? Well, this is the trihybrid cross. And while we could use a Punnett square, you're about to see why that's a really bad idea.
I mean, seriously, a Punnett square for a trihybrid cross would need 64 boxes. 64! That's not just a pain to draw, it's basically begging to make a simple mistake. But luckily, there is a much more elegant and, frankly, smarter way to solve this. And this is it, the probability method. It's a total game changer. Instead of one giant, messy problem, you just break it down into 3 small, simple monohybrid problems. You find the probability for each trait on its own, and then you just multiply them all together. It's so much better. Let's use a practice question to see just how easy this is. We're crossing 2 parents who are heterozygous for all 3 traits. Our mission, find the chances of getting an offspring who is homozygous recessive for all 3 genes. Okay, let's break it down. We look at each trait one by one. For the A gene, what's the chance of getting AA from an AA times AA cross? We already know this from our first Punnett square. It's 1 in 4. And the same exact logic applies to the B gene and the C gene. The probability of BB is 1/4, and the probability of CC is also 1/4. Now for the easy part, we just multiplied those individual probabilities together. So 1/4 times 1/4 times 1/4 gives us 164. And that's it. That's our answer. The chance of getting that triple recessive offspring is 1 in 64. Way easier than drawing a 64-box Punnett square, right? And just like that, you've leveled up from tracking 1 trait all the way to 3. So let's pull everything we've learned together into a practical toolkit that you can use to solve these problems super fast. Seriously? This slide right here is your ultimate cheat sheet. For any cross between parents who are heterozygous for the traits in question, these are the phenotypic ratios you are always going to get. Memorize these ratios and you'll be a genetics pro. 3:1 for monohybrid, 9:3:3:1 for dihybrid, and that big, beautiful pattern for the trihybrid. So we've spent all this time learning the really neat, predictable rules of genetics. But that leaves us with a really fascinating question. What happens when genes decide not to play by those rules? In our next video, we'll talk about pleiotropy and gene interaction. Goodbye.